Good morning, everyone, and welcome to the 2016 Royal Tyrrell Museum Speaker Series. This week, we are lucky to have not one, but two presentations in the context of the Speaker Series, one today and then one tomorrow at the same time. Today, the Royal Tyrrell Museum and its cooperating society are proud to present our very own Miss Calla Carbone. Calla is a preparation technician here at the Royal Tyrrell Museum. Calla is originally from Ottawa in Ontario. She received a bachelor's degree from the Department of Geological Sciences and Geological Engineering at Queen's University in Kingston, Ontario. As an undergraduate student, Calla studied the paleoecology of Ediacaran body fossils from the Bona Vista Peninsula in Newfoundland. After completing her bachelor's degree, Calla decided to stay at Queen's University to complete a master's degree. For her thesis, she studied the evolution of feeding behavior of the Ediacaran fauna by looking at trace fossils found in the Mackenzie Mountains in the Northwestern Territories. Over the years, Calla has volunteered for various researchers collecting Ediacaran and Burgess Shale type fossils and cataloging the latter at the Royal Ontario Museum, has worked for the Ontario Geological Survey, and has even had the opportunity to go backpacking through Europe. Finally, in the spring of 2015, Calla was hired by the Royal Tyrrell Museum as a preparation technician. Today, Calla will talk about her work on fossils of the Ediacaran fauna, the first multicellular animals to have evolved on Earth roughly 600 million years ago. So, without further delay, I present you Miss Calla Carbone.
Hello everybody and welcome. Today I'm going to be talking to you about the Ediacaran period, which is a really exciting slice of the history of life. As you can see from this picture in the background here, these are some of the, what we think some of these Ediacaran creatures looked like, which I'm going to call the Ediacaran biota. And if you take a close look at them, you'll see that they look quite unusual and not really like creatures that we see in the modern oceans. So because of that, this is a very, like a new frontier in paleontology. But in order to actually talk about them, I need to give you some context in the history of life. So if you actually look at this picture here, it's kind of an unusual way of showing geologic time. If you look at the top at 12 o'clock, this is kind of the beginning of the Earth, 4.5 billion years ago or 4.6 billion years ago. And as you go clockwise around the circle, time is getting younger. As you look at the slices of time, these are basically names that geologists have given to geologic time. And when you take a look, you'll see that the bars on the outside basically represent significant new things that have evolved, so significant parts of the history of life. As you can see up in the red up here, that's where dinosaurs, wind dinosaurs lived. And if you look up here, you can't even really see a bar. That's kind of the first hominins or the first early humans. Now, a very common question that researchers are trying to answer is kind of, when did the world begin to look like what it does today? When did the first animals form or evolve? And that question is kind of debatable, but we'll just take a look at kind of early life. So several billion years ago, we have the evolution of single-celled creatures, smaller than you can see to the naked eye, and that's represented by this purple and dark blue line here. And so to give you an idea of what that looked like, or the Precambrian looked like, it looked a little bit like this. So you can see from this picture, it was quite a hostile environment. You have volcanoes spewing gases into the atmosphere. There's actually almost no oxygen in the environment or in the oceans. So pretty much the only thing that could live at this time were microbial colonies that were growing as these mounds in the ocean right here. So we call those stromatolites. Now for a really long time, we thought that the next big event in life was 451 million years ago, the Cambrian Explosion. And that's because that's kind of when we start to see creatures forming or evolving that kind of resemble things that we see in the modern oceans today. You can see there's kind of a worm in the foreground, we see sponges, and some larger predators and things swimming in the ocean. So these for a long time represented the first animals or early animals. But this sudden jump from microbial worlds to complex ecosystems like this didn't really make a lot of sense, and it really puzzled a lot of geologists and paleontologists for a very long time. Even Charles Darwin in 1859 noted that this didn't really make sense. It didn't really jive with his theory of evolution, but he was hopeful that we'd be able to kind of resolve this discrepancy because we hadn't really studied that much of the world at that time. And true to what he believed, about 15 years later, a Canadian paleontologist named Elkanah Billings, you can see right here, found these unusual disc structures in an outcrop in downtown St. John's, Newfoundland. You can actually go to the outcrop today. It's part of a parking lot. It's part of the wall of a parking lot. You can just go and look at these disc fossils. But although we now realize that this is actually a very significant event in the history of life, at the time people looked at these discs and went, those probably weren't living. Those probably are made by some random sedimentary structure. So they kind of just ignored them. And for 85 years, even though they found a couple more unusual fossils, kind of like this disc called Aspidella here, people kind of didn't realize that they were important. So it wasn't actually until 1958 that the first definitely Ediacaran fossil was described. And it was actually found by a young schoolboy hiking through the forest in England. So he found this specific fossil right here, and you can see it has these geometric shapes and a very distinct pattern. And that's how we know that this was created by something that was living. This is a fossil of something that was organic, and they named it Charnia masoni. So now this kind of changes our perspective on life. It actually was a stepping stone that led to the next half century or more of research into what we now call the Ediacaran period. But before I can talk too much more about them, I need to tell you a little bit about what the Earth looked like at that time. So right before the Ediacaran, the Earth was actually undergoing a series of really large-scale glaciations. They called these the Snowball Earth or Slushball Earth. And researchers don't really agree on how much of the globe was covered in ice, but they do agree that it was probably the largest scale glaciation in the history of the Earth. And by the beginning of the Ediacaran, these glaciers were beginning to recede, the ocean was opening up, and this had a really important byproduct. Now the continents were feeding sediment and nutrients into the rivers, which are feeding that into the oceans. And so those single-celled creatures I was talking to you about before, things like phytoplankton, which already existed at this time, were feeding on these nutrients and then photosynthesizing. So that basically means that all of a sudden we have oxygen in the ocean. And just like you and me, most animals need oxygen to live. So this was actually what people think was the reason why the Ediacaran creatures were able to evolve. This is when life got big. It was the spark that allowed life to evolve. Now, since the discovery of Charnia, we found many, many more fossils and many more localities. So each of these dots actually represents a new locality that's been found. And you'll actually see that they're from almost every single continent, basically except Antarctica. And you'll also notice that there are quite a few in Canada. And this is kind of an example of what some of these early finds, fossil finds, kind of looked like. They all have different names now, but at the time when we hadn't described them, we gave them very simple names, like the feather duster, the frond, the comb, the spindle, and my personal favorite, the pizza disc. And you'll notice that these look very different from things like dinosaurs. They're preserved very differently. That's because they're soft-bodied. They have no skeleton. So they're actually preserved as impressions or two-dimensional, almost footprints on the seafloor, which are now preserved as rocks. And one of the hardest things that paleontologists had to do at the time, and still kind of struggle with today, is figuring out what these creatures actually looked like when they were alive. This is an early interpretation of what we thought the Ediacaran used to look like. It's actually a diorama that's still at the Smithsonian. You can see in the foreground we have these jellyfish. Now this did make a lot of sense at the time, based on what we knew about these fossils. But it turns out to be wrong. This is because at the time it made sense to try to compare every new fossil we found to something that was living today. So they thought these Aspidella discs were jellyfish and the Charnia frond was a sea pen. And you can see there is a resemblance, but the closer you look at these fossils, the more you realize that they are very, very different. Some examples of this is, as you can see, there are this cluster of Aspidella discs, and the way that they're kind of clustering together, it looks like they're actually growing on the seafloor and kind of meeting each other, as opposed to a jellyfish that would have died and then fallen from the water column and been preserved on the seafloor. We also see no tentacles preserved, which is another curiosity. But if they're not jellyfish and sea pens, what could they possibly be? Are they plants? Are they fungi? Are they lichens? Are they animals? Are they none of these things? Are they an early evolution of these things? Are they even all related? So it's important to remember that even though all of these creatures are quite unusual, they could have formed an ecosystem that's just as diverse as we see in the modern world. So they might not actually all be related. One of the main overarching questions that researchers are still struggling with today is, were they early versions of an existing group or ancestors of an existing group that we see today, or they failed experiments, which just means a group that evolved and then went completely extinct with no descendants? And to answer this question, we really need to take a much, much closer look at these fossils, and not just at individual morphologies or the different general shapes of these creatures. We actually need to get quite creative and look indirectly at different evidence to kind of infer what these creatures were. And one of the best places to do that is actually in Newfoundland, Canada. So this picture here is a picture of what Mistaken Point, Newfoundland looks like. It's the southernmost tip of the Avalon Peninsula. So St. John's is right here, and the Avalon, the southernmost tip, is basically right there. And so this coastline is covered, has these really large bedding planes that are tilted. And so each of these bedding planes, these rock layers, represent what the seafloor looked like at that time. And this is me standing on one of these seafloors, and you can see they're completely covered in Ediacaran impression fossils. This is such a significant fossil location that it's actually currently being looked at as a UNESCO World Heritage Site. So let's just take a closer look at some of these fossils. How were they really preserved? I said that they're impressions, but they're actually preserved under these layers of ash. So this gray stuff here is actually preserved ash. And it allows them to have a very, very fine detail in the preservation. You can see this is a spindle, this is a spindle, this is a spindle, and this is one of those feather dusters. But this preservation tells us that these were all preserved exactly how they were living at that time. So it's kind of like an Ediacaran version of Pompeii. And this is really useful for us because we can really learn a lot about these communities, because we can see a large group of fossils that were all living together and interacting, and see that snapshot in time. Maybe? There we go. All right. So this is what encompasses the majority of the Ediacaran fossils from Mistaken Point. They're part of a group called the Rangiomorphs. And what that really means is they're creatures that have a series of modules. So you see, this is one creature, and this is one module. And that module looks exactly like the adult version of the creature, the full version of the creature. And then each of those modules is composed of a series of smaller modules, which look exactly like the full creature. So this is like self-similar fractal branching. And this is what some of these creatures looked like. And you'll see that this type of body plan of self-similar fractal branching is primitive, but it does allow for a lot of different shapes to form. So this is the spindle here again. This is kind of a bush-like form, or like a cabbage form. This is the comb again. And then we have 3 different types of fronds. But even looking at these pictures here, it's really hard, I understand, to look and realize what they really looked like. So I'm going to show you a couple of reconstructions of what some of these fronds looked like, done by the paleo artist Peter Trussler. And so you see, this is what Beotheuchus, this is a fossil preserved of Beotheuchus, and this is what we believe it actually looked like. And this is the same for Avalifractus. This is what we think it looked like. So you can see here that even within the frond shape, there can be a lot of variation. You also might notice that these things look quite unusual when you try to compare them to things that we see today. So, what else can we learn from Mistaken Point? What other things can we figure out based on the rocks there? Well, one of them is what kind of environment were they living in? And to do that, we actually couldn't look at the fossils. We had to look at the rocks. Geologists have studied these rocks in great detail and have determined them to be laid down or deposited in water that is greater than 1 kilometer in depth. So that's quite deep where these fossils were living. Now, in the ocean, light actually only penetrates to a maximum of 200 meters. And most photosynthetic things lived well above that or well shallower than that. So this is actually the biggest reason why we know that they are not plants. They could absolutely not have been photosynthetic. And if you look at this picture, this is another rendition of what they looked like by Peter Trussler. And this posed a problem for him when trying to reconstruct what they looked like. How do you make a picture of something that is accurate of things that you wouldn't be able to see because it's too dark? And he actually had to write and explain when publishing this picture that the light in this picture is from a submarine that's shining light. So I thought that was kind of interesting. Now, I've already said that they were, or I should have said that these creatures were all living attached to the seafloor. They weren't really moving. So how were they feeding? How did they eat? Well, if you measure every single fossil on a single surface, you'll actually see that they seem to tear based on their height. And this is something that we see in modern day, things like plants. But if they were tearing like plants, we would expect the tallest tier to be the densest tier, just like the canopy of a forest. However, in this situation, the tallest tier is actually the shortest tier. And this is actually a lot more similar to modern day filter feeding organisms or animals, things that are actually straining particles out of the ocean and then eating them. But, so maybe they're animals, but even the best preserved Ediacaran fossil shows no evidence of some kind of organ like a stomach or something to digest these particles. So the most popular theory at the moment is that these creatures were osmotrophs, which means that they were passively absorbing dissolved organic carbon from the seawater. And that actually fits with the chemistry of the ocean that we know from this time. And that was also probably why they were branching fractally, increasing their surface area to be able to absorb as much nutrients as possible. Because we have these large surfaces, we can also see possibly how they interacted. Now, some of these surfaces are very diverse, have lots of different types on them. And some of the surfaces are almost exclusively one type. Like this surface here, this is only a small portion of the surface, but it's almost all spindles. And I've kind of outlined them because they're a little bit hard to see. But it was actually 98% spindle. And what this tells us is that things like the spindle or Fractofusus were likely early colonizers. Kind of like after a forest fire, blueberry bushes are one of the first things to crop up everywhere. These were like immature communities. And as you increase the diversity or the different numbers of different types of creatures, the community is becoming more mature. These surfaces also can tell us a lot about how they grew and how they reproduced, partially by looking at lots and lots and lots of examples of the same type. So we know that they actually grew in two different ways. One of those ways is by inflation of these modules. And we can tell that because if you look at all different sizes of the same type, so let's look at Fractifusus, for example, right here. Every single one has the same number of modules or the same number of parts to it. And so that suggests that it was inflating while it was growing. In contrast, some of them grew by the addition of these modules. So you can see this is Charnia again down here, one of those fronds, and it keeps adding to the tip to get bigger and bigger and bigger. And for a long time, we thought that these creatures, these rangiomorphs, were reproducing kind of like corals do today. It's called dispersal by waterborne propagules, but basically what that means is they would grow a bud and release that bud into the water column for it to kind of land somewhere else and grow a new creature. But we don't have a lot of evidence for that in the rock record, so it's more of a theory. Recently, however, actually as just last year, a paper was published looking at the spatial relationships between them. So like how far apart were these different fossils and did they form patterns? And they realized that Fractifusus, once again, one of the most important fossils of my talk, these spindles, seemed to have clusters around adult forms. So the largest ones had clusters of intermediate ones, and then those had clusters of small ones around that. And if you look at things today, you'll see it's very similar to stolen asexual reproduction. And what that means is an adult will grow a vine or a stem, and at a certain distance, that stem will root, and it'll grow a new creature at the end. So this is a very similar type of patterning that we see. So it's possible that they actually reproduced in two different ways. Now, I've been talking a lot about Mistaken Point, and Mistaken Point is a great place to study these fossils, but it only really shows one picture of what's going on. This was deep water, and this is one of the earliest, these are the earliest fossils to have evolved, or creatures to have evolved. So to get a better idea of the whole picture, we need to go to some of the other continents and look at the fossils there. And as you'll see in a second, we get a lot of different answers when we go to different places. This is what we call the White Sea assemblage. Mistaken Point is referred to as the Avalon assemblage. And it's best known from Australia, but it can be found elsewhere. And you'll notice that these do not look very much like the rangiomorphs that I talked about before. While the rangiomorphs are definitely not plants, are probably not animals because they're not filter feeding, instead they're absorbing nutrients. These creatures look an awful lot like bilaterian animals we see today. So things with bilateral symmetry, a definite head, a definite tail. So we think these might be the first definite animals, early animals. And these creatures were probably also moving on the seafloor. They weren't attached to the seafloor like the rangiomorphs. And this White Sea assemblage is from rocks that are interpreted to be much shallower in depth than the rangiomorphs assemblages. Now if we can compare this to rocks from Namibia in Africa, we see a whole new assemblage called the Namus assemblage. So these are entirely different creatures once again. This group is called the Ernietomorphs, and they are basically a series of tubes that are quilted together to form various body plans. If you look at the stamps, they're kind of small in the corner here, but you see kind of what we think they looked like. So one of them actually kind of looks like a frond, but it's a very different type of body shape or body pattern. We also see this little guy here, Claudina, which is the first Ediacaran fossil that has a more rigid skeleton. I wouldn't say that it's not a fully fledged skeleton, it's just more rigid than everything else. And it's a series of nested cups. These guys have been found to form reefs at this time. Now, I've talked about water depth, so it's easy to assume that the different assemblages just mean different environments, where the mistaken point assemblage is from the deepest water, the White Sea or Ediacara assemblage is from shallower water, and then the Namus assemblage is even shallower than that. But unfortunately, it is not that simple. We also have to think about time. So this diagram basically shows the different appearances of the different Ediacaran fossils. So blue are the Rangiomorphs, yellow are the Ernietomorphs, orange are the bilateral forms, and then there are quite a few other groups of Ediacarans that I'm not going to talk about today. And then if you plot them against time, so we're getting younger as we go up here's the Cambrian, you see that the Avalon assemblage at mistaken point is both deeper and older. And then the Namus assemblage at the top is both shallower and younger. So it's hard to figure out whether these assemblages are different because new things are evolving, or if they're different just because they're from different water depths. And if you think that's complicated, we'll take it one step further. It's even more complicated than that, and that's because of the way these things are preserved. Now, I talked about mistaken point being preserved under these ash layers, but that's not the only way these creatures are preserved. Each of these assemblages are preserved in different ways, and they're preserving different parts of the same body, and sometimes different organisms entirely. And to give you an idea of what I'm trying to say here, is we'll look at Aspidella again, so the disc. We've actually come to realize that this disc is actually just the base of a frond. So it's the holdfast that's tethering fronds to the seafloor. And the reason we know this is because if you have preservation from the bottom, all you see is a disc. But if the frond gets pushed over and you have preservation from the top, like these ash layers, you'll see the whole frond. So the same organism looks different, but the same creature looks different based on the different ways that it's preserved. One of the really important ways that these are preserved is because of the microbial mats on the seafloor. So I said they're attached to the seafloor, but this seafloor looked really, really different from the seafloor we see today. It was covered in microbial colonies that formed these large sheets, like mats. So that's why I call them microbial mats. And they actually help to preserve these fossils. Now, luckily, this whole problem of is it evolution, is it environment, or is it preservation is slowly being figured out. And that's because we keep finding new fossils, and new fossils that don't really fit with what I've said to you already. So all of these things are things that have been found very recently. In the last 2 years. This is a frond that's preserved in that three-dimensional Namus-style preservation. This is an Ernietomorph preserved in deep water. This is another Ediacaran fossil that's actually preserved in a very different type of rock. So it's a limestone instead of a mudstone or a shale. And then here we actually have a new taxon, so a new creature. It kind of looks like Cloudina. It has that rigid shape, and it looks like a tube, but we've called this Sequitubulus annulatus. So this is helping us untangle what was going on at that time. And so this is a little bit of a scary picture. It's a scary diagram. It's a cladogram. But what I want you to get from this is what we've kind of learned. We've kind of learned what they are. So this tree of life is showing branches of the sponges, the cnidarians or corals, deuterostomes, which are basically vertebrates, and protostomes, which are the invertebrates. And as you can see, all of these four groups are part of animals. And if you take one step closer, avoid the sponges, it's a subgroup of the animals. And then if you get rid of the corals, we have these two groups, the invertebrates and vertebrates, that are bilaterian, so they have bilateral symmetry. And if you plot the Ediacaran fossils on here, you'll see the Rangiomorphs fall somewhere off to the side. So we think these are failed experiments. They don't have any descendants. They evolved and then completely disappeared. They're not animals. However, a lot of these early bilaterians of the Ediacaran are probably exactly that, early animals. So these are the first glimpses of animals that we see. Now, these bilaterians, like bilaterians that we see today, or even us, were actually able to move, unlike these rangiomorphs I was talking about earlier. And these are two examples of this evidence of motion. We call them trace fossils. These kind of circles form kind of trails, and at the end we see a specific type of fossil called Dickinsonia. And what this suggests to us is that these are feeding tracks where Dickinsonia was crawling on the seafloor, would stop, and then absorb the microbial mat underneath it. And here's another example. This is Cambriella. It's kind of like an early mollusk-like creature. We thought it had a proboscis that would scratch the microbial mat for it to feed. Now, these represent two of the body fossils, creatures that we have preserved in the rock record, making traces. But there's a whole other group of early animals that have no body fossil record. We only see the trace fossils, so these burrows preserved. And that's because they're too small. They're millimeters in size. And the best place to go and study those is the Mackenzie Mountains in the Northwest Territories, so in Canada again. So these creatures were feeding on the microbial mats, creating really, really small burrows that are preserved on slabs of rock like this. So as you can see, like I said, millimeter scale. Because it kind of looks like it's going in and out of the plane, each burrow kind of will go from positive to negative relief. We think that it was feeding in the top couple of millimeters of a microbial mat. And then you'll also notice that they kind of appear aimless. They're grazing burrows of microbial mats that don't really seem to care where they're going. And this is really significant because it will tell us a lot about the behavior of these moving creatures. So at first they start off aimless, but as time gets younger, we start seeing what this arrow is, a behavior where a burrow will come close to a previous burrow and then turn away and move away from it. We call this phobotaxis, but basically it's just avoidance behavior. They are purposely avoiding previously burrowed areas. And this example is just one example. It looks pretty coincidental. You can kind of just brush it off. But as you go to younger rocks, still in the Ediacaran, you have more and more evidence, and it appears more and more deliberate. So you can see there are actually 3 areas where you see avoidance behavior, and it actually looks like this creature is beginning to form a meander. And that's important because the youngest rocks that are still Ediacaran have these actually quite complex meandering patterns to them, quite tight meanders. And this process of moving really close to a previous burrow, but parallel to it, so not crossing it, we call thigmotaxis. But really, what does this say? We're getting more patterned behavior. Well, this is actually showing that these creatures are getting smarter. They're beginning to actually have organized ways of feeding. This is when sensory systems were beginning to evolve, so things that eventually led to brains today. And sure, it's kind of hard to think, oh, well, patterned burrows mean things are getting smarter. But the reason why this is happening is the same as when you're trying to plow a field or harvest a crop. If you looked at this field and tried to just make a random path all around it, it would take way longer to plow it or to harvest it. It would waste so much more gas, and you'd probably have patches that you've missed. So in the same way, by forming a complex meandering pattern, you're actually maximizing the amount of food that you're going to come in contact with and minimizing the amount of energy that you're wasting, basically. And we actually see this behavior in the deep water today of creatures that are burrowing in sediment that is nutrient poor, so there's not much food there. So basically what's going on at this time is about 600 million years ago, we have Ediacaran fossils. About 555 million years ago, these burrowing animals appear. And then over time, they begin to outpace the growth of these microbial mats because they're eating them faster than they can grow. And then by the end of the Ediacaran, there are these kind of patches of microbial mats where Ediacaran fossils are still living. And then these areas that with a more modern substrate, kind of it's aerated, it's sandy or silty or muddy, where new things are beginning to evolve, new feeding behaviors and such. It's because there's a new ecological niche opening up, like a new habitat. And you do see this in the rock record. So this here is just time, and we're getting younger as we go up. The Cambrian boundary is about here. And these are the different feeding strategies that we see from the trace fossils. These are some of the examples of what they kind of look like. And you just see there's a stepwise increase in the diversity of behaviors, from just microbial grazing to the addition of deposit feeding, predation, and suspension feeding. And we can see that all from these trace fossils, these burrows. So to sum all of that up, the Ediacaran is actually quite a complex ecosystem. There were early animals, there were things that completely went extinct, but this ecosystem, these creatures dominated 20 million years of life. So quite a considerable chunk of time, these creatures were dominant. And at their peak, there were creatures that were attached to the seafloor, feeding on dissolved organic carbon. There were creatures scuttling around on the seafloor, eating microbial mats. But then we get to the Cambrian, and there's so many things that have changed. We have new creatures, we have things that are predating other things, we have skeletons forming, the ocean chemistry is changing, and these Ediacaran fossils disappear. So what happened? Well, there are three theories as to why the Ediacaran biota disappears. The first one is fairly simple. It just means that they didn't actually disappear, but the microbial mats that were preserving them disappear because they're being fed on by the burrowing creatures. So it's just a disappearance of the preservational window. The second theory is that there was like a dramatic catastrophic extinction. So something was going on either in the chemistry of the water or something was going on basically that caused them all to just completely die out. And if that were to happen, we'd expect a lot of chemical signatures in the rocks, which do happen, but we would also expect them to disappear quite quickly. And there's quite a gradual disappearance in the Ediacaran biota as we get closer and closer to the Cambrian. So it's unlikely a catastrophic extinction. The third theory is biotic replacement. And that basically just means that other creatures were outcompeting the Ediacarans. So the evolution of these early animals that were burrowing and eating the microbial mats were kind of destroying the habitat that the Ediacarans were living on. So these Ediacaran fossils were probably or creatures were falling over because they couldn't attach to the seafloor properly anymore. And in addition to that, we have things that are evolving that are actually eating other creatures with the evidence of predation and biomineralization. But so if something is going to eat something else, the easiest thing to eat is something that doesn't move and is soft-bodied. So the most likely scenario is that it's actually biotic replacement. But either way, whatever the reason, these creatures disappear about 541 million years ago. At least that's what we think. Now, this is not definitive, but I'm just going to kind of throw this out here. In 2014, researchers were dredging the deep ocean and found these creatures. They found these creatures here called Dendrogramax, and they've actually believed that they're animals, but we don't really know anything else about them. They could be a new phylum, we don't really know. These here are some Ediacaran fossils that I have not discussed today. They're discs, but they're not holdfasts. They have radial symmetry. Here it's triradial symmetry. And then they have these networks features coming out here, these little branching patterns, very similar to what we see here. So it is possible that some of the Ediacarans, or the strictly Ediacarans, did survive into the modern. So we've come a long way looking at Ediacaran fossils from the first discovery of Charnia. We've done quite a lot. We now have an idea of what was going on, what the creatures were living on, how they were reproducing, what they were eating. We have a pretty good picture, and we have an idea of what we think that they were. But there's still a lot of work to be done. A lot of these big questions, even what were they, are still a little bit unresolved and are pretty heatedly debated in the researching community even today. So lucky for us, we find new fossils every year, and this kind of makes the picture a little bit clearer every time we find a new fossil. So who knows what fossil we'll find next and what that will actually mean for researching in the Ediacaran. With that, I'd like to say thank you to the Royal Tyrrell Museum for letting me talk today. Thanks to Queen's University and everyone involved in helping me write my master's. And thank you for listening.